Hello children, in this video we are going to learn differences between antigens and antibodies in 7 different points. While studying biology we come across many terms, some of them which are somewhat similar to each other. Like here 4 terms are mentioned: antigen, antibodies, antibiotics and antiseptics. All these 4 terms have anti as their prefix, but all these 4 have different meanings. They are all different from each other. In this video, we are going to discuss only the differences between the first 2 terms. First difference is based on synonym. Synonym means another name of the term. So antigens are also known as the immunogens. Immuno means immunity and gens means generating. Antigens are immunity generating substances or materials, whereas antibodies, they are known as immunoglobulins. Globulins is a kind of protein, so antibodies are immunity developing proteins. Second difference based on definition. Antigens are foreign material that enter our bodies and bring about a reaction in the body. As soon as they enter our bodies, a chemical reaction begins. Okay, whereas antibodies are a biochemical which are formed in the body to fight the antigens. Third point is based on purpose. What is the purpose or use of the antigens? What do they do? So, they produce disease and allergic reactions in the body. And apart from this, they trigger the formation of antibodies. Whereas antibodies are protective in nature. They kill the antigens by various methods. Various methods like they may bring about their agglutination or they may cause their lysis. Fourth difference based on their origin. Antigens come from external environment or from within the body also, as we see in the case of autoimmunity. And the antigens may be free molecules or they may be components of surface covering of the microbes. Microbes which enter our body like bacteria, fungi, amoeba, virus, etc. Whereas antibodies are produced within the body, always within the body, and they extrude from the surface of plasma cells and they become a component of the blood plasma. Fifth difference, that is based on the molecular structure. Antigens may be proteins, polysaccharides, lipids or they may be nucleic acids also, whereas antibodies are always proteins. Sixth point, antigens have specific binding sites which are known as epitope. Epitope means it is a part of antigen which can be recognized by the antibodies. This will become clear in the next point. And paratope in the case of antibodies, this is the specific binding site of antibodies, a part of antibody which recognizes and binds to the antigen. Now look at the next point, the 7th point, the shape. Shape and examples also we will discuss here. Now see here, antigens can be of various shapes, like they can be a protein, complex protein or a simple kind of protein or a nucleic acid, or they can be a bacteria or amoeba or a virus, fungi, etc. And I have drawn here another antigen of this particular shape because it can be of various shapes. And a particular antibody always recognizes a specific antigen only. Now look at the shape of the antibodies. You can see that they are usually always Y-shaped. And antibodies, you can see that it has a combination of 2 light polypeptide chains and 2 heavy polypeptide chains. Light polypeptide chains means less number of amino acids are there. Heavy polypeptide chain means large number of amino acids are there. Now these chains, they are kept together or held together with the help of the disulfide bonds, as we can see here. Now this part of the antibody, this is known as the variable part, and actually this is the paratope. The antigen recognizing part of the antibody. Now look at this antigen. It is having a specific shape, and this area, this part is the epitope. Now this is a combination. This antibody can recognize this particular antigen only. This antibody cannot recognize these other antigens. Now when it recognizes this particular antigen, this will come over here and it will fix over here like this. It will bind, and now it is the turn of the antibody. It can kill the antigen. It can kill the antigen by various methods. Finally comes the examples. As many times I took the name of all these antigens examples in this video. So once again, it can be by bacteria, virus, protozoa, fungi, or it can be free molecules like fats, proteins, nucleic acids, etc. And these antigens, when they enter the body, they cause diseases or they trigger the immunity response. Now it is the job of the antibodies to attack these antigens and kill them, and do not allow them to cause any disease in the body. Antibodies are of 5 different classes. As you can see here, they are called immunoglobulins A. As you can see, students, the immunoglobulins A, they are found in all the body secretions. They are found in mother's milk. They are found in colostrum, the first milk of the mother, which is very important for the newly born child. Second immunoglobulin, that is immunoglobulin D. It is found in small quantities in the blood serum. The third category, that is immunoglobulin E. It acts as a mediator in the allergic responses. The fourth category, immunoglobulin G. This constitutes 75% of antibodies in our body. It is found in all the parts of body, blood, lymph, intestines, and in the mother's milk also. And finally, the immunoglobulin M, which is the largest of the antibody. It is found less in number, but it is the first to reach the site of infection. So this is how, when antigens enter the body, it is the job of the antibodies which are produced within our body to fight with them, kill them, and protect the body from different kind of diseases. I hope you liked the video. Thank you all.